Good day, STEM student. This is Teacher Jan, your biology teacher. For today, we are now in Biology 1, Week 5. In our previous lesson, we learned about cell division. So let's have a review. In doing our review, we are going to play Who Am I? Are you ready? Okay, for the direction. Guess what is mentioned in the riddle? You only have 30 seconds to answer each item. Okay, let's start. Time's up, so the answer is cell cycle. So let's have another one. Time's up, so the answer is mitosis. So let's have another one. Time's up, so the answer is meiosis. Great job everyone, you got all the correct answer. For our new topic for today, it is all about cell cycle malfunction, the disorders and diseases. So now, are you ready to enter the spectacular journey of exploring the effects of cell cycle malfunction? Okay, let's go! But before we proceed further, let's first establish the learning objectives. Upon completing the module, the learner should be able to remember the regulatory mechanisms that control the cell cycle, identify the sources of errors in the cell cycle, and define disorders and diseases that results from the malfunction in the cell cycle. As we go through to our discussion, you will notice this unfamiliar word, so remember this. For our trivia for today, did you know that the female produces a finite number of egg cells in its lifetime? These egg cells takes 45 years to mature and some may die before it matures. The male sperm cells, on the other hand, are being produced all the time. This is the reason why risk of birth defects are high for females at older age. Now, let's recall the checkpoint in cell cycle. There are several checkpoints in the cell cycle that maintain the integrity of the process. The G1 and the G2 checkpoints ensure that the cell have enough nutrients to stand on its own, while the M checkpoint ensures that each cell receives the same amount of chromosomes or two chromosomes have separated equally. The cell cycle is prone to errors due to the phases of genetic variation that occurs in meiosis and the DNA replication process in mitosis. When the cell is unable to correct the damage made at this point, it goes into a programmed cell death, also known as apoptosis. These are the possible sites of error. 1. The DNA replication, the process by which the genome's DNA is copied in the cell. 2. The recombination, a process by which pieces of DNA are broken and recombined to produce a new combination of alleles. And 3. The independent assortment. Genes independently separate from one another when reproductive cells develop. When these regulatory mechanisms are unable to pause the cycle and the cell fails to go into apoptosis, the abnormal cell will proceed to multiply. So now, we will be discussing the disorders caused by a malfunction of the cell cycle. Let us first take a look on how a normal human chromosomes look like just before nuclear division. Take note of the number of chromosomes present in a cell. Chromosomes 23 is the sex chromosomes, which dictates if the cell will grow into a female or male. Let's talk about the non-disjunction disorders. It occurs when either the homologous chromosome or sister chromatids are not able to segregate correctly. This results in daughter cells having an abnormal amount of chromosomes. The two most common effects of non-disjunction is the aneuploidy and polyploidy. Aneuploidy has an abnormal number of chromosomes, lacking or having an extra number of chromosomes, while polyploidy has an abnormal set of chromosomes. One of the examples of aneuploidy is the Down syndrome. It is a chromosomal disorder which occurs when an individual has full or partial extra copy of chromosome 21 due to errors in cell division. The extra copy of chromosomes 21 impairs the cognitive ability and physical growth of the child. Trisomy 21 is the most common type of defect which causes Down syndrome. During cell division, chromosome 21 of either parent cell failed to separate, thus, one daughter cell received 3 copies of chromosomes while the other received only 1. The effects of Down syndrome are delays in development, intellectual disabilities, slanted eyes and shortened neck, short stature, and poor muscle tone. Mosaicism is a type of Down syndrome where the cell presence in a baby is a mixture of cells with 46 and 47 chromosomes as shown in the picture. Those cells with 47 chromosomes contain an extra copy of chromosome 21. This mosaicism is the least common form of Down syndrome. In translocation, there are also 3 copies of chromosome 21 present in the cell. The extra copy of the chromosome is attached to the other chromosome instead of being separated. Another example of trisomy disorder under aneuploidy is Patau syndrome, also known as trisomy 13. It is due to a person having 3 copies of chromosome 13. Patau syndrome is a serious condition because it may result in either stillbirth, miscarriage, or infant death. Here are the effects of Patau syndrome: low birth size and weight, holoprosencephaly, abnormalities in face and head, and problem in baby's organ. Another example of aneuploidy is the Edwards syndrome, or also known as trisomy 18. It is a very severe genetic condition that affects how your child's body develops and grows. Children diagnosed with trisomy 18 have low birth weight, multiple defects, and defining physical characteristics. Another is a Turner syndrome. A normal female has X chromosome while a male has X and Y sex chromosomes. The female receives its X chromosome from both parents. A Turner syndrome occurs if she fails to receive both X chromosome. This error happens during the formation of the egg or sperm from the parent cells. It is a condition that affects female only. A child with Turner syndrome have short hair, congenital heart defects, spatial awareness tissue, and absence of menstruation and hearing problem. Down syndrome, Edwards syndrome, and Patau syndrome are examples of autosome. Human autosome are chromosome pairs of 1 to 22 chromosomes, which are the non-sex chromosome. Triploid syndrome is an example of polyploidy. It is a rare condition where a cell has 69 total chromosomes instead of 46. This genetic abnormality affects fetus and is life-threatening. The condition causes several effects. Those are abnormal brain development, adrenal and kidney defects, widely spaced eye and low nasal bridge, low set malformed ears, small jaw, absent or small eye, and cleft lip palate. The last disease that we will be discussing is leukemia. It is a type of cancer usually known as blood cancer. It is a condition where defective white blood cell crowd the red blood cell and platelet in the body. When the cell fails to go into apoptosis, the side effects of leukemia include anemia, excessive bleeding, hair loss, and sore mouth. Since we have finished discussing disorder and diseases,
Let's engage in an activity. Are you familiar with a game called Among Us? The mechanics of this game involve three characters introducing themselves. They are Mr. Red, Miss Pink, and Mr. Blue. Among them, two are impostors, and your task is to identify which one are not the impostor. Are you ready? Okay, let's start. You only have 30 seconds to answer. There are many diseases which are caused by defective cell cycle mechanism. But the most common disorder and diseases we discuss in this lesson, in aneuploidy we have Down syndrome, trisomy 21. Under Down syndrome is the mosaicism and translocation. Next is Patau syndrome, which is trisomy 13. Edwards syndrome, trisomy 18. And Turner syndrome 45X. In polyploidy, we have triploid syndrome 69. And the last disease is leukemia. For your assessment for today, answer the 5-item quiz. Good luck, everyone! That's all for today. See you in our next class. Bye!